let's get into germ theory a bit. It is a big part of the book, and we've talked about germ theory a lot since COVID. Like I mentioned in the beginning, sometimes there's a big trigger event that makes you look at something a little more deeply than you ever did before. And COVID and germ theory, that was it for me. And I don't know if people really think much about the details of germ theory and viruses. Like the things that really set me off were we tend to think about bacteria and viruses as kind of the same, but they're very different. Viruses are 10 to 1,000 times smaller than bacteria and can only be seen with an electron microscope. And that's far different from bacteria. If you look at the scale of microscopes and what they can see, like if you just do a Google image search, they'll show you the scale of small things and the microscopes needed to see them. Yeah, you need a special electron microscope, which is not really available to most people. And when you really start to think about, is this thing that is like 1,000 times smaller than the naked eye the cause of my illness? Does that make sense? My body's pretty powerful. Is it really crippling me that it's this little tiny thing? And also, I had a previous guest mention that viruses, they don't even know if they're alive or dead. And I thought that was very strange. And I looked it up. It is true. If you Google it, are viruses living? The result I got was most biologists say no. Viruses are not made out of cells. They can't keep themselves in a stable state. They don't grow and they can't make their own energy. Even though they definitely replicate and adapt to their environment, viruses are more like androids than living organisms. That is what Google says is the official word. So they're so certain that viruses are the cause of 99% of our problems. Nobody can see them without the special equipment and the lab coat mafia. And they don't know if they're alive or dead. That seems like a pretty fundamental thing to know. You know they're the cause of all our problems, but you don't know if they're alive or dead? That seems pretty insane.
Yeah, actually, they say they're not living. Like you said, that's the official stance. They talk about something, when they talk about one being alive or not, they talk about being replicant competent or not, meaning can it replicate, as in DNA replicating. Like they see it, it's sort of modeled off of like what they say DNA is, or RNA. So viruses are supposed to be like an RNA or DNA encapsulated in a protein shell. And that's how they describe them. However, this might be a big news to a lot of people, even though they're supposed to be able to view them with an electron microscope, nobody has ever actually viewed them. What they have viewed is what they assume to be virus particles. Now, if viruses existed in the first place, so there is a question whether they exist or not, you would be able to see them with the electron microscope. But no scientist to date has seen them except for Dr. Stefan Lanka, who is a German virologist. He was able to isolate and view sea algae viruses. And what he thought were, so they have these ones called like, what they call them, like not big viruses, but something like like some other word that's like a larger virus, like he thought he was seeing this like bigger kind of viruses. And he was like, oh, look, I found this like bigger kind, right? And the ones in humans and animals are supposed to be smaller, especially the pathogenic ones that they talk about. And so what he found out was then he did more research and he found out that nobody had ever actually isolated and identified and viewed these so-called pathogenic viruses that are supposed to exist all around us and in humans and plants and animals. Nobody's ever done that. So the idea of a virus, which actually means poison in Latin. So viruses exist and that poisons exist. But what happened was germ theory was kind of falling apart because, there's a lot of problems with germ theory, but one of them is that at some point they didn't find, so sometimes you can find bacteria that, present when certain disease symptoms are present. So with tuberculosis, there's a tuberculous like bacteria that was actually like characterized and identified and mostly viewed in people suffering from tuberculosis, not always. Sometimes people had diagnosis and symptoms of tuberculosis without these bacteria present. and some, so it's never been consistent with the bacteria being present with the disease symptoms. But because whenever it happened at all, they just assumed that, okay, that's what's causing it. The assumption's amazing to me. We see these bacteria present, they must have caused this disease, and it's not even consistently present every time with these disease symptoms. Right. So how do you actually they've never proved pathogenicity with any of these. They've never proven germ theory, and I can get into that. But the point is, at some time, they found diseases where there were no bacteria present that they could point to as the boogeyman. So they said, well, it must be something smaller that we can't see yet. We must just not have the equipment to see it yet, right. Which is insane, because there's always going to be something we can't see. There's always going to be that. There's always going to be. So what do you just assume it's something there that we can't see that acts like this with, that aligns with germ theory? Right. So that's what they did.
Rather than abandoning the premise, it's just no, the premise is right. We're just going to have to look harder.
Exactly. And so they said, well, there must be something smaller. So they said, oh, well, they came up with this idea of viruses. And that was in 1915. So not that long ago. And it wasn't until 1930 or 35 that they had the electron microscope and that they could finally view viruses with. And guess what? They didn't. So they did not view them. And this is a shock to a lot of people, but nobody has seen them. So, but they're assumed to exist, but they know all about them because of computer models, right? And so just a lot of assumptions, no real science in terms of proving germ theory. The only proofs that have been conducted are usually involving torturing animals, favorite thing to do, torture animals. So, what's interesting to me is the isolation process of what they call isolation is a total bastardization of the term. It's better now, if you say purification, if you're trying to talk to scientists about this, it's better to use the word purification because they've just completely bastardized isolation. So, because we think of isolation as you take the one thing from the other thing, the surrounding material. So you have somebody's spit who you think is sick and suffering with this virus, and you're supposed to spin it around and what's it called? Centrifuge gradation. Yeah. And then get your little pipette and try to separate that out, so that way everything's separated from each other. You separate the virus out and then you view it. You put it on your slide and whatever. But they've never done that because they can't find them actually. And so what they do instead, and this was Dr. Peebles came up with this in the 1950s, a surrogate kind of way of testing for viruses and isolating them or proving that they exist, doing the same thing to prove that both they exist and that they cause disease. And what they've done is they take, say, you're sick, you spit up some mucus for them or your pee, it doesn't matter, or some blood sample, and they'll take that and they'll put it in. So they have a culture of a tissue culture, and that's what you're going to put this into, right? And the tissue culture comes from ground up monkey kidneys, is the one way, monkey kidney cells. So, it has to be the green, the rhesus monkeys, certain monkeys, they imprison them, torture them, rip out their kidneys and grind them up in a blender. And then you have that, or you can get it from eggs. Or bovine serum, that's a fun one. It's aborted calves, take that stuff. And so that's the tissue culture. And then they take the spit from the sick person, and the idea is you're going to take that and put it into this culture, cell culture, and see if it kills off the cells or not. And if it does, then you're going to assume that there's a germ in there doing that. You're not going to see the germ, but you're just going to assume it's there, and you're going to assume there's a virus there. So there's a lot of assumptions again. And the problem is they never do a control, meaning they never take spit from a healthy person and do the same test and see if the same thing happened. So because there's all these things they do in between these steps, and what they do is all these samples are completely destroyed through chemical processes. So they'll take that tissue sample, has to be fresh because the cells are immediately going to be dying. And so what they do is they first add bactericides to it and other chemicals to make sure that everything else, if there are germs in there, they're dead. And they never just test to see if just doing that to the sample is killing it. So They also put these things in incubators, heat them up, and they let them sit for a couple days. So what they'll do is they'll look at the student, and there's all these instructions, and I have it in the book. Like the student looks through the microscope at the tissue sample, and they say, okay, I see cells living, and they set it aside. And then they add the bactericide, and then they do all these other processes to it. They don't look at it again after that. They only wait until they also there's a bunch of processes that the sputum goes through. And then they put that in the culture, they let it sit for 2 days, and then they'll go back and look under the microscope. If a certain percentage of the cells are dead, you have both isolated your virus and proven that it's pathogenic. That is what passes as science today for virology. I mean, it's insane. It's actually crazy. No controls done.
Can you believe it, people? A video clip and a peek into how that sweet THC sausage is made. For some people, I'm sure it's nice to put a face to the name, so I started making clips that are a little more YouTube friendly than the full show tends to be. Get the full show on any podcasting platform. I prefer to keep it audio only, so we have a more decentralized distribution for a controversial show, and also so I can edit out the ums and ahs and barking dogs and all the things that happen when a person records in their home environment. And then if you really like the show, you can sign up for THC Plus, and instead of 1 hour interviews, you get the full 2-hour interview for just $8. I think that's a better deal, more of a win-win than asking you to support the show by booking a therapy session or buying generic Viagra. It's getting weird out there, folks. So just go to the highersidechats.com or click the link at the top of any set of show notes, and all your wildest dreams will come true. All right? All right.
The Higherside Chat Show, Greg Carwood and Company.